great.
Yes, we can advance.
Okay.
And I'll introduce you. The camera doesn't work, but then
I
the
saw
two
your,
minutes
yeah.
Okay, the next company we'll be hearing from is Athersys, and presenting for them today is Bob Deans, Executive Vice President, Regenerative Medicine.
Yeah, that's great. Thanks. So I've been very happy at Athersys. I joined Athersys in about 2003 and just have really had a fine time developing out the adherent stem cell technology. And it's really been fun for me to work with Gil, and particularly in the last 3 to 4 years, see how much impact his energy and enthusiasm have had on the field overall. So I say over that all together, but I'm going to prove it to you because I've actually agreed to give a talk using his slides. So what greater commitment can a guy have? So Athersys is a publicly traded company located in Cleveland, Ohio. We work with an adherent stem cell derived from bone marrow, and I'm happy offline to talk to you all about its distinctive features and the methods we have for isolation and expansion. We've been very committed to developing a knowledge-based technology platform. We've worked very hard both with cell therapy societies and industry organizations to promote openness in publication of preclinical results, and also proactively published our clinical trial design and obviously promote publication of clinical data. So I'm going to go through some of the details of the cell product in terms of its value as a universal cell product linked to its expansion capability and its immunological features. And I'm going to show you where we stand in our portfolio of indications. And one of our early models was to build out a core competency in isolating and expanding this cell population, and then to target physician networks that we thought could be well served with going after indications with significant unmet need, and then target pharma and healthcare providers that had interest in that space and could serve as co-development partners and as a distribution partner. And so in that sense, we had a very broad approach in the beginning, and it's actually helped us quite a bit. We've learned a lot from the bench to bedside and back to bench approach about how the cells work mechanistically. And even though we've been in some diverse therapeutic areas, we've gotten an understanding of the common pathways that our cells use in those settings. So our entry point was actually in using the cells to treat inflammatory injury in allogeneic bone marrow transplant. And at the time we filed our IND around 2004, 2005, this was really where adherent stem cell classes were going to the FDA to get approval for manufacturing, to get a general biosafety approval. And we were able to execute a phase 1 study using the cells delivered at the time of hematopoietic transplant to demonstrate a reduction in incidence and severity of graft-versus-host disease. We've had an orphan status granted around that, but most importantly, our experience in that phase 1 study was able to leverage us into a related indication. The common immune pathology in gut, in inflammatory bowel disease, and in graft-versus-host disease enabled us to go forward to the FDA and enter directly a phase 2 study in ulcerative colitis. And our experience in GVHD was able to convince a partner, Pfizer Regenerative Medicine, to serve as a co-development partner there. So it was a very successful execution for us. We've also been able to demonstrate that these cells impact regulatory T cell populations, have preclinical data that has supported an investigator-sponsored IND using the cells to promote engraftment in solid organ transplant. We hope to have that study started in Germany before the end of the year. We've been very active in the cardiovascular space. We have a phase 1 study in acute MI that recently closed. We've had a very durable effect there with an average improvement in ejection fraction in our top 2 dose tiers of between 8 and 10%. That was durable, as I mentioned, out to 12 months, and we've published that data in Circulation and actually made the front cover of Circulation. And we've had backup studies in CLI and Ph.D., and we've gone to the FDA, and again, they've told us that we can enter directly at a phase 2 stage in studies that look at benefit provided by angiogenesis. And so some of our more exciting recent work has been in the CNS area. We have a very nice pipeline of activities here that's been led by ischemic stroke. And, to beat a dead horse here, we've also been able to go directly into a phase 2 study in ischemic stroke based upon our biosafety experience in infusing the cells at high dose. I'll talk more specifically about the stroke study, and then I'll give you some more information about the other CNS indications. So I think very many people are aware with the MSC-like platform with adherent cell technology, and I'm not going to go through that extensively. We do have a more potent expansion potential for the cell that lets us go through a master cell bank and working cell bank approach in manufacturing. It has a benefit from our perspective in letting us have uniform clinical doses be used in treatment. So we've been able to use the same clinical master cell bank in all of the clinical indications I've indicated. And from a single bank, we can get into the hundreds of thousands of doses, a dose for us being about 200 million cells. I think you're also all aware that these cells function as delivery vehicles for biologics. They're not cells that have an active therapeutic role by regenerating tissue. And we've been very careful to study the multiple pathways through which they work. And it's very important to reemphasize the point made in one of the early panels that this is not a single modality of action, but these cells are working through multiple pathways in parallel. And so for where we've analyzed these pathways in vitro, we've been able to verify that they're active by extensive in vivo tissue microarray analysis. And we've been able to develop potency assays and affirm these mechanistic pathways in vivo by doing knockdown and add-back experiments. So we have a really deep data set around how the cells are working. And so I had mentioned that we had entered the therapeutic space with our GVHD study, and then went on to an alliance with Pfizer Regenerative Medicine around inflammatory bowel disease. And we chose specifically to go into the ulcerative colitis population. We were concerned in this space about having a very clear quantitative endpoint, and so our endoscopy and Barron score are really our clinical endpoints. This is a dose crossover study that had an early stage of an early stage of 2 dose tiers being tested before moving to the highest dose tier. I think one of the things that I want to, and this is a description of the study design there, one of the things that I would point out is that we've been doing well with our product delivery and patient accrual. We're targeting complete accrual of the study somewhere in the mid-2013 time space, and that's going to represent the first proof of mechanism data for us coming out of a phase 2 study that we hold very important to continued development. And then this was really just a summary of our phase 1 GVHD study. We've just completed the clinical manuscript and are submitting it for review, so soon you'll be able to see that as well. One of the things we've learned here is that the cells appear to be acting by suppressing inflammatory injury due to the myoablative conditioning. And as well, we've been able to demonstrate in vitro and in vivo that our cells block homeostatic T cell expansion. And this is the propensity of donor T cells when delivered into the myoablated patient to undergo cytokine-driven expansion to fill the secondary lymphoid organs. And so the take-home message for us, which has been corroborated by our clinical analysis and preclinical study, is that by limiting tissue damage and limiting the ability of the T cells entering the body post-transplant to amplify, we've reduced the probability that we're going to initiate an allogeneic sensitization response. And again, I'm happy to talk about that with you all at any point during the meeting. So our best, a really sweet pipeline of activity has been in the CNS space. And I think over the last 3 to 4 years in looking at partnering activity in stroke and getting a better understanding of the risk aversion that pharma has, given all of the failures in the stroke space, we've learned some important lessons. And again, those lessons are understand how the cells work, get a strong rationale for your dose regimen clinically, and just keep to your guns, stay with your convictions where you believe and understand the technology. But we've been able to gain significant foundation or grant support for the rest of the pipeline. We've had $1.5 million STTR, SBIR in both traumatic brain injury and neonatal HI. The Third Frontier program is enabling our IND-enabling studies to go into the clinic in spinal cord. We've had very good support from Fast Forward in developing our MS program. And so I think it's been a very effective and economical way for us to build a very good therapeutic pipeline. I'm going to show you on the next slide a model for how we understand these cells are working in stroke and TBI, and it represents words that are actually on this page, and I'm just going to go to the picture. So what we learned is that our cells are having a very significant influence on the systemic immune response, and the systemic immune response is acting and causative for many of the pathologies in stroke. And so we find that when our cells are put in anywhere from day 2 to day 7 post-injury, they have the consequence of shutting down the blood-brain barrier. They shift inflammatory cells from an inflammatory phenotype to a wound repair phenotype, a Th1 to Th2 type. They shift activated microglia and macrophages from a phagocytic to a repair process. And they're doing that by both changing the circulating inflammatory cytokine profile from TNF-alpha interferon gamma to IL-10, IL-4, and we're inducing a population of regulatory T cells that are going into the brain and again are limiting neuronal cell death. They're cytoprotective, and they're shifting the polarity of the activated inflammatory cells to a repair process. And so this has been a very important theme that's coming out in several of the last recent stroke meetings, that people understand the very tight linkage between the systemic immune response and the pathology seen in stroke. And I think we're all aware and probably have had family members that have suffered from stroke, and we're aware that there really isn't any effective treatment out there, that the thrombolytic properties of TPA are effective, but many people cannot receive treatment within that very short required window of time. So we also believe in taking strong phase 2 proof of mechanism studies forward. And so we have a dose escalation study in stroke that has 114 patients to be accrued. I mentioned earlier we went through a dose group of 400 million cells blinded and matched with placebo as for safety evaluation, and we moved up to 1.2 billion cells as an effective dose. Our safety committee has reviewed the data and recommended going forward with the high dose. So we're now at the stage of rolling product out for the third cohort, which is the paired randomized blinded high-dose treatment study. So this is going to be our second POM study. It's going to come out somewhere, it'll accrue somewhere end of 2013, early 2014. So we're looking at a horizon of somewhere between 15 to 18 months to have some significant clinical data to report out and help advance the platform. So we've done very well in these other studies that I had mentioned in the CNS pipeline. We've been working with Bob Miller at Case Western Reserve and can show functional benefit and have gone through dose tiering and are ready for a GLP pivotal safety study, preclinical study, before moving forward in MS. We've published several studies on our work down at UT Houston in TBI, and we've done very well as well in our spinal cord model. In that setting, we've been able to demonstrate that our cells have a primary impact on the activated macrophage population immediately post-injury, and the consequence of that is to prevent axonal dieback or pullback from the site of injury. We find that we can prevent that by influencing the activated macrophage and stimulate the cells to actually grow through the glial scar. So it's really a remarkable finding. But even more satisfying is we've been able to understand that we accomplished that by limiting the exocytosis of MMP9. So we know very specifically, in a very specific pathway, and we've corroborated by using small molecule inhibitors of MMP9, that we understand how our cells are acting with specific components of the immune complex to affect that recovery benefit. And so this is, I'm surprised I haven't gotten the high sign. Thank you. I'm down to 1. So this is a slide that basically recapitulates the therapeutic areas that we're in, but I've developed kind of a Pavlovian response to the slide. I was able to meet my parents over the weekend this week and going through the standards, so what do you do in your life and how's everything going at work? And the words from my mother keep ringing in my ear like, Bob, when are you ever going to focus? So I think no, I say it because of the love we all have for this therapeutic space and the broad utility of the cells, but I think I'm proud that the company has reached a point where we understand very well how the cells work and we can make an intelligent decision about how to focus our therapeutic pipeline. So thank you.